So tonight is battling bugs, inroads and infectious disease. So if you came here to learn about termites and cockroaches, you might be in the wrong plane. These are little bugs. These are infectious diseases. We have some great speakers lined up for you today. Dr. Brad Cookson. Dr. Cookson is a professor of laboratory medicine and microbiology here at the UW School of Medicine. He's executive vice chair and vice chair for clinical operations in lab medicine here at UW School of Medicine. And he's the head of the clinical microbiology division and director of the molecular microbiology laboratory here in the School of Medicine. He's developed detection systems including next-generation sequencing for diagnosing bacterial, fungal, and parasitic infections with the goal of improving the medical management of patients suffering from infectious diseases. Here he is with a big infectious disease. That's a big bug you got there, Brad. His laboratory discovered a new biological paradigm, a conserved pathway of pro-inflammatory cell death called, you have to get this, pyroptosis, which is evoked by infection and other important pathological insults such as cancer, heart attack, and stroke. He also received a Pfizer New Faculty Scholar Award and chaired the Federation of American Societies for Experimental Biology Research Conference, Microbial Pathogenesis, Mechanisms of Infectious Diseases. He's a great teacher, and his trainees are attending physicians and faculty at academic institutions across the U.S. He earned his MD and Ph.D. degrees at Washington University School of Medicine in St. Louis, not to be confused with the University of Washington. That's the other Washington. And completed his residency in the Department of Lab Medicine here at the University of Washington School of Medicine. He was senior fellow in the UW Department of Immunology and the Howard Hughes Medical Institute.
Good evening, everyone. Thank you very much for being here. It's my pleasure to have the opportunity to speak with you. I'm impressed that all these people are willing to forego the Olympics to come listen to John and Paul and myself speak tonight. So this evening, I thought I would begin with a historical perspective about infectious diseases with a quote from Sir William Osler, who is widely regarded as the father of modern medicine in the Western world. And he said that humanity has but three great enemies: fever, famine, and war. Of these, by far the greatest, by far the most terrible, is fever. And so what I'd like to discuss with you this evening is three stories developed here at UW Medicine to help deal with this terrible situation of fever: a traditional approach, a molecular approach, and a next-generation approach. Fever is, in fact, a host response, and at a molecular level or cellular level, fever results from inflammation. And the reason it's important to think about inflammation is because inflammation very often indicates the presence of a problem. The significance of inflammation in human disease is more widely appreciated than it once was. And importantly, inflammation is not limited to infectious agents. Other important causes of disease, in fact, the four leading causes of diseases in developed countries: heart disease, stroke, cancer, as well as infection, all share an underlying mechanism of inflammation. So that leads us to the important consideration that we can only solve problems that we know about, and the best way to solve a specific problem is to have a specific solution to that problem. And so effectively, a treatment that's going to give you a cure depends on diagnosis, which comes from the Greek roots, or has Greek roots, and it means the determination of the nature of a disease. And its synonym, diakrisis, comes from the Greek root of through, a judgment. So, for an effective diagnosis, one public service announcement I can provide this evening is that Sir William Osler also provided us with some sage advice for us as patients, because your physician, if they listen to you, will hear from you key details about the diagnosis. And for you as a patient, that means to be an accurate historian, you can impact the way your health is delivered or cared for in our delivery system. So, a diagnosis is really a scientific approach of hypothesis testing. So you're going to come into the doctor with a problem. Some of them, Dr. Lynch outlined, are going to be dramatic. And your problem is one that the doctor is going to ask you, well, when did it start? What makes it better? What makes it worse? Where have you been lately? And according to Dr. Lynch's talk, they might also want to know who you went with and what you did when you were there. All right? And from that, your physician is going to formulate a hypothesis, which then they can test with a physical exam. And they're going to use their skills of listening or auscultation with a stethoscope on your heart and lungs, percussion on your chest, and palpation, say to make sure that your abdomen is soft and non-tender. And at that point, the physician is going to modify the hypothesis and engage in the process of testing that hypothesis, potentially with imaging or laboratory testing. And laboratory testing is the domain of laboratory medicine. And possibly none of you knew that there was a branch of medicine focused in the laboratory. And so I thought I would take just a couple of minutes to explain to you what it is. The Department of Laboratory Medicine has expertise in all aspects of human physiology. For example, chemistry of cellular functions. Are the cells alive, intact, doing what they're supposed to do? Are they producing what they need to produce? Are the organs that these cells form, the liver, the kidneys, the pancreas, all producing the important molecules that you need to have a healthy and happy life? Hematology, that's the study of those red blood cells that carry oxygen to your tissues, the white blood cells that fight off infection, and transfusion medicine is about making sure that the tank is mostly full most of the time. You need red blood cells to survive. Immunology. Immunology is very important because that is an entire system devoted to defending your body. So is that important? I ask the audience for their participation. It is. We're a Super Bowl town now, and we realize that defense wins championships, and the immune system is all about having a great defense system. And it defends you against pathogens that are described in the microbiology or the virology labs. And there are also individuals with expertise in medical informatics, which is conveying this data to the physicians who have engaged in the scientific process of hypothesis testing so that they can narrow down the problem and focus the treatment so that you live a long and happy and prosperous life. And one aspect that we're going to discuss this evening is part of the molecular diagnosis program that's run by Professor John Tate in the Department of Lab Medicine. And I'm going to tell you about one laboratory there, the Molecular Microbiology Laboratory. And this is a collection of talented individuals, and I just thought that I would mention that a lot of this work is conducted by our staff. We're second only to the School of Nursing in terms of or nursing at our hospital in terms of total staff. There are almost 900 people in our department. And one way that you can enter into this process, there are physicians such as myself that are board certified in pathology, board certified in internal medicine or pediatrics with subspecialty training in transfusion medicine or rheumatology or infectious diseases. There are also doctors with Ph.D.s that are board certified by the American Board of Clinical Chemistry or the American Board of Medical Microbiology. But then there are a lot of talented staff that are medical laboratory scientists. That's a 4-year degree that you can get at the UW, and they're certified by the American Society of Clinical Pathology. They perform the procedures and operate instrumentation that provide physicians with the data in order to make a diagnosis and affect a quality treatment. So why go to the trouble of identifying an agent that's causing infection? We want to determine the nature of the disease, and very importantly, we want to predict the course and potential outcomes of infection, because whenever you get sick, let's face it, everybody wants mommy, and that's because mommy always gives great supportive care, and supportive care is really important for combating disease. It also provides us with the opportunity to tailor therapy. You'll hear more about that from my colleague, Dr. Pottinger, but as Dr. Lynch pointed out, that is an extremely important part of our current medical care system that we need to pay great attention to. And it's also important, as I pointed out earlier, that there are many causes of inflammation. We want to exclude non-infectious causes for your symptoms. There are infectious agents that are very, very clever. They're called the great imitators. The most experienced clinicians have mistaken an infection for another process because these agents are capable of infecting pretty much any organ in your body. And so that's the nature of their ability to be a great imitator. They imitate many different diseases. And so I thought it would be also time for just a quote from me, which is, the reason you want to identify these etiologic agents is because what you can't see can in fact kill you, all right? So let's do everything we can when you're a patient. Please be a good historian. And also, let's take the Super Bowl approach. Let's have a good defense. Valentine's Day is coming up. Go home to somebody that you love and say, I want to make sure your vaccines are up to date, because defense wins championships. You know, the Seahawks are helping us out here in a way that I'm very grateful for. Okay, the identification of etiologic agents. The traditional approach is, if it grows, we can identify what it is. And that would mean we would acquire a specimen from the patient. That is most often a fluid, blood, or urine, or cerebral spinal fluid. And that fluid is going to be subjected to microscopic exam. And here you can see a little snippet. It shows us a couple of important things. These cells are polymorphonuclear neutrophils, or PMNs. They're primary guardians of our body, and they respond rapidly in the 6 to 24-hour range at the initiation and onset of infection. And also, we can also see here, as a result of the Gram stain technology developed by Hans Christian Gram in the late 1800s, gram-positive cocci clusters. And so now what we would like to do, or the strategy is to isolate these microbes individually and then amplify them so that we can identify what they are. So it's a little bit like drinking at a fire hose when you get a specimen from the leg of the patient that Dr. Lynch described. And you can see over in this portion of the plate that there are a large number of colonies. And as you swirl this way, they get fewer and fewer until we have individual colonies. That's important because I took a picture of this plate yesterday. There are 5 different organisms there, and we can't specifically identify all 5 at once with this culture-based approach. But what we can do, we can utilize the phenotypic attributes of these organisms. And what you can see here is that, for example, this is on agar that contains red blood cells. You can see a discoloration around this colony, and it's because this organism is producing something that's causing lysis or partial lysis of the red blood cells. And you can see that's very different than this colony, both morphologically as well as the surrounding media. And then you can see very interesting things like this organism right here, these concentric rings of so-called swarming motility. This organism is motile, and we can use a constellation of the morphological features of these organisms growing on media, as well as a variety of biochemical properties, that is, enzymes that they produce to identify what they are. And for example, this organism is likely a member of the genus Proteus, and a single enzymatic reaction allows us to provide for Dr. Pottinger and Dr. Lynch information that distinguishes one version of Proteus that's susceptible, let's say, to ampicillin and cephalosporins from a different version of this same bug that would be resistant to those antibiotics. So we can use these morphological features, the phenotype of the organism, if you will, as well as its biochemical attributes to identify what the organism is. And the reason that's important is we take this information, both the phenotype and the gene products that we're capable of analyzing in the laboratory, to provide us with an identification. And the reason identification is important is because of we want to tailor the therapy, and we want to know the natural history or course of disease and potential outcomes. Remember, that's the mommy response. We want supportive care. The other reason it's very important, you could say, Dr. Cookson, why not just establish what antibiotic resistance mechanisms are present and what antibiotics would be useful for treatment? Well, it turns out that that's an informed process that is greatly aided by identifying the microorganism. The other important aspect that's part of healthcare is that with the identification of the organism, it may lead to additional therapies for other people, for example, that you've been exposed to, or an epidemiologic investigation, or as Dr. Lynch pointed out, this patient is going to remain in isolation because they're infected with organisms that are a great risk to everyone else in the population, whether you're hospitalized or not. This is wonderful. This is a wonderful opportunity. We know that the phenotype, which is the physical expression of the genotype of the organism, and these gene products are responsible for the phenotypes that we see. So the proteins we can often detect with reagents that allow us to identify what the organism is. The presence of enzymes, for example, I pointed that out just a moment ago. And they also make complex structures that we can detect. They literally, bacteria have appendages, just like my arm with my hand, that allow them to adhere to the surface of cells, for example, in the urinary tract. Okay? Imagine, it's difficult to swim upstream, but imagine trying to hold on in the flood. Okay? E. coli has the capacity to do that. And all of these attributes of the microorganisms, the phenotype and the gene products that they produce, are really as a result of the expression of their blueprint or their genotype. Now, that leads us to a new situation where we need to address the shortcomings of the traditional approach, because here I've emphasized if it grows, we can identify it, but what if it grows very slowly? Does anyone know of an infectious disease where it's better to treat it slowly or treat it next week as opposed to today? No. Time is very, very important to give you the best possible outcome. And what if this organism doesn't grow in the lab at all? What if it's disguised its usual characteristics? And importantly, what if it's never been seen before? We've described here at UW Medicine new agents that have not been seen before. And now, with the appreciation that these organisms cause disease, they're recognized in patients all over the world. So really we have to temper our enthusiasm for the traditional approach with slightly different emphasis now, and that is, if it grows, it can usually be identified. So what are we going to do in this situation? Again, it's important to identify the etiologic agents of infection so that we can tailor therapy, establish the natural course of illness, provide the best possible care. One opportunity we have is to interrogate the genotype of the organism. So this approach is, again, very similar to the one we described before, which is to isolate and amplify. Okay? When we get a colony growing up on a plate, we have a single bacterium that is going to amplify 10 to the 9th times, a billion-fold. And in a billion-fold amplification, we'll get a colony on a plate. Now, there is a very nice website that I refer you to that if you Google scale of universe and look at powers of 10, it will give you an appreciation for what a billion-fold really means, okay? This film was developed in 1977 for IBM, and it begins with a picture of a man and a woman on a picnic blanket in a park in Chicago, viewed from 1 meter. And by the time you get to 10 to the 9th meters away, not only are you far from the Earth, but the Earth is no bigger than a marble in your view. So 10 to the 9th is a billion is a large number. So what is our approach? Our approach would be that each of the individual colonies on that plate would contain a species-identifying DNA sequence that, if we can amplify it using conserved primer binding sites, in other words, this is a handle for us to get access to your Social Security number, right? There's this indicates one flavor of bacteria, if you will, a different flavor of bacteria in fuchsia, and a different flavor here in the darker purple. So, if we have the opportunity then to interrogate these isolated organisms, look at their Social Security number, their DNA sequences, we can identify them, tailor antimicrobial therapy. But the whole reason we need this approach is that they may not grow in the lab. And that's where, again, we need to utilize the process of amplification. And so, having identified this target for which we can use DNA sequencing to identify the microorganism, we can amplify it through a process called the polymerase chain reaction, where 2 copies of the target become 4 copies, and when we copy each of those, we get 8, and when we copy each of 8 copies, we get 16, and when we copy each of 16, we get 32, we copy each of 32 copies, we get 64, thank you. So not everybody's watching the Olympics on their smartphone. And so we can use this process to get 10 to the 9th amplification, and this allows us to interrogate those specimens for microorganisms that we cannot cultivate in the lab. And the impact for the patient is turnaround time with a diagnosis, a treatment plan, appropriate antimicrobials, avoiding the promulgation of resistance, and another Super Bowl Sunday where you're sitting in the stands. Perfect. Okay, our first case is a 79-year-old gentleman, had a soft tissue excision. In other words, there was a mass excised. On microscopic evaluation, there was lots of inflammation. But there were no microbial elements, no bacteria seen, no fungi seen, no parasitic elements seen. And all of the cultures were negative. So the question is, could this gentleman have an infection where antibiotics would be the cure, as opposed to possibly treating him for something else? All right? As often happens here at UW Medicine, our physicians are clever, and in this patient's history, they carefully wrote that the differential diagnosis included an infectious process, possibly a blank infection. They're hot on the trail in this case. It could be an autoimmune inflammatory process, such as rheumatoid arthritis or a rheumatoid nodule. It could also potentially be a neoplastic process. Again, this all follows from our understanding that inflammation underlies many different possible diseases. And in this case, this gentleman had inflammation, but there wasn't evidence of infection. So we conducted amplification and DNA sequencing on a paraffin-embedded formalin-fixed tissue specimen from this patient. And in this case, we identified mycobacterial infection. Now, the physician initially the team initially thought that this could be a mycobacterial infection, but we were able to provide additional information. This was an infection caused by Mycobacterium avium. And the therapeutic approach to that disease is different than it is to Mycobacterium tuberculosis. And the more specific therapy we provide for you, the sooner you get a great outcome. Case number 2. This is a case that was brought to us by Dr. Joe Zunt in the Department of Neurology and worked on by a former fellow in our program, Amanda Harrington, who's now an assistant professor at the University of Illinois, Chicago, and she directs their clinical microbiology lab there. This gentleman had refractory seizures. You can see that this is an MRI, cross-section through his cranium like this, and you can see that there's edema in the right parietal lobe that is enhancing with gadolinium, which is an indicator of inflammation. Lots of serological tests were done. That's where you send a specimen of the patient's blood and ask, are they making an immune response to a particular pathogen? The cultures were all negative, and in this case, the patient elected to have the lesion surgically excised. The microscopic examination of that lesion showed mixed inflammatory cellular infiltrate, and again, there were no microbial elements present. Again, we used the amplification process in DNA sequencing on a fresh surgical specimen, and we identified Tinea solium. So the diagnosis in this case, which is consistent with the patient's eating habits and travel history. Tinea solium is the pork tapeworm. It's contracted by humans by eating undercooked pork products. And the diagnosis in this case was neurocysticercosis. Okay, this last case is from a little girl who had acute myelogenous leukemia, and this is an example of her lymph node biopsy using a special silver stain. And you can see here that there are formed microbial elements, and at least to my microbiologist eyes, the pseudohyphae, it's very beautiful structure. It did not grow in culture, but for a person that's immunocompromised with leukemia, this fungal infection represents a threat to her life. And so again, we performed an amplification process in DNA sequencing to identify that this organism allowed a specific antimicrobial therapy, because with fungal agents, as my colleagues will tell you, the difference in cost of a daily dose could be between a few dollars and a few $1,000s. And getting it right is the difference between being alive tomorrow and being dead tomorrow. And so in this case, this patient had an infection with Candida albicans, which is good news because it's very treatable and it's also does not require the most expensive antifungals. Okay, so full disclosure, the genomic approach also has its shortcomings. And the shortcomings are that what if the infection occurs in a site with normal microbiota? And what if the infection occurs and has more than one pathogen present? So the key to success with the traditional approach and the genomic approach is that we isolate the organisms and amplify them such that we can query them one at a time and assess their identity through DNA sequencing or morphological features one at a time. So the situation exists where what if all three, orange, fuchsia, and burgundy organisms are present at one time? So Dr. Lynch mentioned the concept of microbiota, and really you are a super organism. That is, there's the cells which are derived from your mother and father's genetic union, and then there are also the cells, we all have baggage, psychological, emotional, but everyone for sure has microbial baggage, and you have it in lots of different places. And the relationship we have with that microbiota is a relationship we can call, it's mutualistic, okay? They derive benefit, we derive benefit. Did you know that when you shave this morning, your legs, your neck, I guess maybe face, maybe some of you have to shave your back, I don't know, that your ability to coagulate depends on microbiota, because that vitamin K is important for gamma carboxylation of coagulation factors that allows coagulation to occur. Okay, so if there's infection present at any one of these sites, there's the presence of many different templates and many different organisms, and that is going to make even our fancy genomic approach difficult to reach success. The other potential situation, which is known to occur, is that there are many sites in the body where polymicrobial infections are common. So it exists in a variety of areas. One example would be in an abscess where there's more than one organism present in that situation. And in that case, it's going to be challenging with the genomic approaches and as well as the traditional approaches to isolate a single organism, identify it so that we can then prescribe the appropriate antimicrobial therapy. So what then is the approach? And I was inspired back in the early 90s by something that a female physician had said to me once, that I thought, you know, there's going to be a time and a place where I'm going to be able to use this information, and I had to wait a long time, but this evening is the opportunity I've been waiting for. And the physician was Beverly Crusher, assisting Dr. Toby Russell in Star Trek The Next Generation, episode number 115. Now, I don't know if you're familiar at all with Star Trek The Next Generation, but Lieutenant Worf was the chief security officer aboard the Starship Enterprise. Again, defense wins championship. So Lieutenant Worf's functionality was very important. So Beverly Crusher and Toby Russell, it turns out Worf was in a cargo bay and had a tragic accident. He was going to be quadriplegic, and they engaged in this process called genetronic replication of his spinal column. And in order to complete that process, they were sequencing the genome at 10 to the 9th base pairs per second. It turns out that we're not quite there yet, but certainly when I was a fellow here, I was sequencing 400 base pairs in 4 days. So we have come a long ways, and this is my opportunity to say thank you to all of you. Our government has a national agenda to fund research, and a lot of the sequencing technology that has impacted medicine today is a result of that national research agenda. So you can pat yourselves on the back for that. Thank you very much. So the investment in sequencing technology now allows us to penetrate this space where there are multiple microorganisms. And so how is that going to work? This is the next generation sequencing approach. Again, we're going to isolate and we're going to amplify. And realize now, I'm going to propose to you that we're going to go headlong into the fire hose and begin drinking and receive nourishment and great satisfaction. Now what we're going to do is we capture single DNA molecules, and in the same way we grew single bacteria into a colony on a plate, we're going to amplify a single molecule into about 1,000 copies of that molecule, and we're going to call that a cluster. And then we can sequence each of those clusters. And it turns out we can sequence 15 million of those clusters in parallel. I want to say that again. A single molecule, we're going to isolate millions of single molecules. Each of those are going to be amplified into a small cluster, and we're going to sequence all of them simultaneously, potentially 200 million at a time on a large cartridge. This is a small cartridge. I took this picture yesterday. That's my quarter. That's our cartridge. It's a single flow cell cartridge, and there we capture single molecules, and we can sequence millions of them at a time. So why is this useful? This allows us to peer into the human body in places and gather information that we never have been able to gather before. Boldly go where no one has been before. I believe at this point Kirk would say, brace for impact. I would go like this a couple of times, and all of you would slam against that wall and slam against that wall. Okay, I mixed Star Trek generations there, and I apologize for that. What does it allow us to do? Bacterial vaginosis is a medical condition for women where the unfriendly bacteria are growing in their vagina. This is a contribution from my colleague Dave Fredericks at Fred Hutchinson Cancer Research Institute. And the important point here is that the patients down here with green bars do not have bacterial vaginosis, and they have a very homogeneous, lactobacilli-rich vaginal microbiota. The patients suffering from bacterial vaginosis, indicated up here in these columns with red, have a very heterogeneous microbiota in their vagina. Now, Dr. Fredericks has not been able to ascertain which of these organisms is causal, but certainly it provides a great deal of information about the difference between those healthy patients and those suffering from bacterial vaginosis. So stay tuned here at UW-Medicine for more information on that. This is a study done by Noah Hoffman, who's an assistant professor in laboratory medicine with a very capable laboratory medicine resident, Steve Salaponte. And you can see at the bottom that the traditional culture-based approach, these are respiratory specimens from patients with cystic fibrosis. The traditional culture-based approach that's shown down here with the colored bars indicate different organisms. The size indicates their abundance. You can see that the cultures from all of these patients appear to be rather similar. When we use the next generation approach, you can see that they actually stratify into 1, 2, 3, 4, 5 different groups. Now, we don't know what that means yet, but we're hopeful that this kind of information is going to help us target therapy for individuals that have different microbiota present at sites of infection. So I'll close with a case, and we did go rocket speed from the earlier part of human history until this is a case that's only a few weeks old. A 35-year-old gentleman with meningitis is found to have a brain abscess. All the cultures are negative. When we did our traditional amplification and DNA sequencing, it revealed polymicrobial infection. And unfortunately, the current standard of that technology does not allow us to comment any further. It turns out that we performed next-generation sequencing on this specimen, and we identified 3 different bacteria that were responsible for the meningitis. The antibiotic regimen was then tailored specifically to that patient's infectious agent. It's important to note that the most expensive antibiotic was excluded from the list. And the most recent information from the physicians caring for this patient, and this is a patient that's not in Washington State, is that he's recovering well and moving on. So, our mission is to support the greatest possible healthcare delivery for our patients, not only those of you who are treated at UW-Medicine, like myself, but for those in our region. And I hope today that I've shared with you some reason why you have great confidence in UW-Medicine. I think that we're doing our best here, and we receive specimens from all over the world. And it's indeed a privilege to be able to provide these services. I went to medical school for a reason, and that's why. It's a privilege to be here. So one last thing for those of you who may have questions, if you Google UW Molecular Microbiology, you can go to our website, and it has interesting information about papers that you might want to look up, clinical significance, and other sites that you can be directed to provide you with additional pieces of information. Thank you so much for your attention.